So in doing DNA typing, there's actually two typing kits. They're called amplification kits that are utilized to come up with the totality of the 13 locations.
Let me ask you, I'm sorry, Ms. Seubert, would it assist you to use the board to kind of explain it a little?
So the areas that we've kind of talked about, there are 13 locations on the DNA, and they are selected because of their highly polymorphic, or they're very different between individuals. And they're actually located on different chromosomes. And we haven't talked about chromosomes, but you actually have 46. 23 of them came from your mother, 23 of them came from your father. And so on those chromosomes, certain locations have been identified as being highly polymorphic or different that have been selected to be looked at in forensic DNA typing. And so within one of the kits, there are actually 9 of those locations that have been identified and that we will amplify the DNA with, or target those locations, make the copies, and generate, if there's enough DNA there, a DNA result. And so I'm going to write out those DNA locations for you so you can see their names, and I can tell you a little bit about what each of those names means.
Can everyone see it? Okay.
So what I've listed out here are each of, this is 9 of the locations that we would look at in one of the tests, or that are contained with one of the kits. And that kit that I've listed here is the Profiler Plus ID kit. And each of these locations, this one is D3S1358. D3 is actually on chromosome number 3. And it is the location, it's basically like its house address on the chromosome. If you think of the street that you live on, it's the main street, and it's kind of like the house number for the area that's been targeted. And so as you go down the line, you have VWA, FGA, D8S1179, D21, I'll shorten them, D18, D5, D13, D7, and then amylogenin, which we've been talking about as the sex typing locus, which will tell whether or not you have male or female DNA present. And so what happens during the DNA typing at the very end, as a part of my analysis, when I do my interpretation, I'm actually looking at the peaks that Mr. Bias talked about earlier at each of these locations. So at D3, I would actually look at if there are peaks present and what the DNA type is for each of those peaks, and then I would populate the table, which would represent the DNA profile. And what these peaks actually represent, and we've talked about is DNA, but these are actually the short tandem repeats. They're STRs, which are the types of DNA repeats that are present at each of these locations. And so essentially counting up how many of those repeats are present at each of the locations that were examined.
And do you get one of those numbers from your mother and one from your father?
That's correct.
Okay. And what happens if both of your parents have the same one at that location?
So if your parents have the same STR type at that location, then you're going to have just the one peak. So for example, let's say we'll call this D3 here, and let's say this is a 1516, and let's say we go to VWA, and there's just one peak there. That means both your mother and father have given you that one type, and then you would just be, we'll call it a 10. And so both parents have given you that 10 as part of your DNA makeup at that location.
So what we've looked at so far that you've written, in a hypothetical situation, this person would be a 1516 at this marker, and then a 10 at this marker.
Sure, and that's, yeah. So each of these markers, there will be if there's enough DNA present to generate a result, there will be a peak or 2 peaks present if you have a single source sample.
And when you have the full combination of the 13, that is when you have a DNA profile.
Well, you can actually have a DNA profile just at one location. So you don't necessarily need all 13 to have a DNA result. It could just be at one locus, it could be at 2, it could be at a combination of them. And so I've only listed out 9 of the locations here. The second test actually has another series of these locations contained within it that are targeted. And that's where you get the additional loci would be in the next test or locations.
And depending on the marker, it tells you how discriminatory it is, meaning how many people might have that number at that location?
That's actually a different part of the process.
Okay. Now, how many of these markers do you need to exclude someone?
It could just take one marker to exclude an individual.
Okay. This is, well, let me ask you this first. Is it possible when you contaminate DNA that it may overwrite DNA that's already there?
Well, it would depend on the quantity of the DNA that you're suggesting is the contaminant. So there would be some factors that would have to be considered with that.
Okay. Explain those factors.
Well, depending on the quantity of DNA, if you have a very large quantity of DNA, it is possible that it could shadow or hide the very small presence of DNA from another individual. Well, any time an item is handled by fewer people, there's a less of a chance of another or of individuals' types being deposited on that item. So sure, the fewer people that handle an item, the less chance there is of a contaminant or another DNA profile being deposited on it. But that still didn't change the way that I would have processed that item.
Now, the elements do and can degrade DNA, does it not?
Overall.
Sure, the elements, sunlight can destroy or deteriorate the DNA that's present, humidity, those several different factors of being outside could impact the amount of DNA that would be left on an item.
Is it possible for you to know whether you'll get DNA from an item unless you actually test it?
Can I answer that outside the yes or no, sir? Well, that would depend. If it's a blood sample that is a red-brown stain, I expect to get a DNA profile from that reddish-brown stain that I've concluded is blood. If it is a general swabbing item that I've not identified any biological staining, but I'm trying to collect biological material, in that case, I don't know if I'm going to generate a DNA profile.